bacteria increases and more of them are around in the general area, or they just get pushed closer and closer together, these autoinducers will start to not only come out of the cell membrane by the bacterial cell that is producing them, but also be able to be transported back in through the membrane. And once this happens, it is able to bind to that blue receptor, that LuxR, and then LuxR subsequently is able to bind to the DNA of the bacteria. And it binds at such a specific position that it turns on or activates the transcription of certain genes related to quorum sensing. So what this means is that bacteria are able to regulate when they turn on certain genes based on how many other bacteria are around them. And so as you'll see on the next slide, these bacteria can do a variety of different things based on whether or not there is a quorum or a certain number that is reached of themselves.
So this is the chart that explains all of this. So some of these acylated homoserine lactones will be paired with multiple different bacteria and they'll have different behaviors. So as you can see, these are, not really ideal, human pathogens, plant pathogens. And the one that we're focused on specifically is the Vibrio fischeri bacteria with the LuxI synthase and the LuxR receptor. And this is because the behavior is really easy to identify, which is bioluminescence. So we're able to test if our inhibitors work or not.
Yep, and then the key thing just to remember is that these spelled something wrong, great. But, but that these all have that same kind of backbone, that acylated homoserine lactone. And then an interesting point that was mentioned is that there's about 20 of these that are known, which means that some of them can be used for interspecies communication and intra-species communication, which is what Emily was kind of alluding to.
Okay, so now there's different ways to inhibit this pathway. So the first way is to inhibit the receptor. So this is basically saying it's like competitive inhibition. So we make our own we make our own autoinducers, and they go through competitive inhibition with the receptor. So if a better generated autoinducer binds, it won't produce any behavior. Like those bad behaviors will be disrupted. So basically the bacterial cells can speak, so they can still produce these AHLs, but these receptors aren't listening. They're not able to receive those autoinducers. So there's a lot of research here. So that's why we work on inhibiting the synthase, because there's not so much research here. So what this means is we're inhibiting the synthase, we're making sure that the signal won't be produced to begin with, and that will inhibit the pathway overall. So these bacterial cells can hear, these receptors can still hear, but there's nothing to hear because these cells can't really speak because they're not making a signal. So they're not making a signal that will produce an undesirable behavior from happening. So that's what we're focusing on. So like we said here, the first way is to inhibit the receptor, and some examples of these inhibitors of the receptor are shown here. And as you can see, some of them kind of mimic the AHLs that we have in this chart here, which makes them ideal receptor inhibitors because they're able to, like I said, inhibit in a competitive way with the not good AHLs. So what's special about these is this one here, the CVIR, is one that Scott made and put in a paper. So shout out to Scott there, because this is on the stars here.
And then me and my covalent bond content. So these two are especially special because they can actually form a covalent bond with an amino acid in the back of the pocket on the receptor. So what happens here is that we have two kind of types, two flavors. Number 22 is a malamide, and number 23 is an isothiocyanate, abbreviated ITC. And so there's something actually really interesting about compound 22, is that if that malamide portion that's in green were actually substituted to be a succinimide, which just means deleting that bottom double bond, making it no longer a kind of conjugate addition acceptor, then it would actually revert to being an agonist instead of a irreversible inhibitor. So if you remove that double bond portion down at the bottom of compound 22, this actually will activate the receptor instead, which is kind of wild.
Huh, that's surprising.
It is very cool. And so there are two ways to kind of tell whether or not these inhibitors actually work. Like we were learning about, these are the assays. So the first way is called a reporter gene assay. So like I mentioned when I was discussing like how we turn on these certain genes with the receptor, if we insert a gene into that little strand of DNA that's transcribed when quorum sensing is reached, that we know the phenotype of, so like maybe it turns green and glows, then we are able to tell whether or not the receptor is bound to the DNA and actually activated transcription. So if we see green and glowing, it means our receptor inhibitor did not inhibit the receptor. It means that it still was able to transcribe the DNA and we see the results. If we don't see green and glowing, then our inhibitor actually worked in inhibiting the receptor and it did not bind to the DNA and subsequently transcribe it. The second type is called EMSA, which means electrophoretic mobility shift assay. And on the next slide, we have a figure supplied by Scott of what this looks like. So EMSA is a way of visualizing whether or not the receptor coordinates with DNA, much like the reporter gene assay. So we can tell at what concentration this potential inhibitor prevents the coordination. So it is a protein gel where you add the DNA that is radio labeled so we can actually see it. So every band you're seeing in this gel is DNA. It's not protein. And so we can tell where the DNA is based on whether or not it is coordinated with our receptor. So if it was coordinated with the receptor, it gets heavier because the receptor is this big old protein. So in lanes 2 and 3, it's heavy. You can see it's at the top of the gel. It doesn't flow through as quickly. So that is when the DNA is bound to the receptor. As you add more and more of our inhibitor, which is the plus CL, then the DNA slowly shifts down towards the bottom of the gel, meaning that is no longer coordinated with that receptor and that our inhibitor actually works in blocking the ability of the receptor to coordinate with that DNA. So it's no longer heavy and the DNA moves further. So those are just two ways of being able to tell whether or not our inhibitor actually does its inhibiting.
So speaking of agonism and antagonism, these graphs here just kind of show what they do in a very general way. So an antagonist is a turner-offer, so it's going to stop a behavior from happening, and an agonist is a turner-oner. It's going to promote a behavior to happen. So in these graphs, if we start out with 0% behavior, this typo, oh, turner-oner, okay, sorry. Okay, so if we start out with 0% behavior and we pipette in an agonist, you'll see an increase in behavior.
You are.
And if we start with a higher percent of the behavior and we pipette in an agonist, you won't really see any change and you'll be able to confirm that you do have an agonist because it's not really changing. Versus if we start with 0% behavior, pipette in an antagonist, you're not going to see any change because there's nothing to stop. If we start with 100% behavior, pipette in an antagonist, you're going to see a decrease in that behavior. So partial agonist or partial antagonist is a mix of both. We start out with 0% behavior, pipette in an agonist, you'll see an increase in behavior up to around 50%. And at the same time, while you start with 100% behavior, you pipette in an antagonist, you will see a decrease in that behavior to around 50% or so. So these curves just show that in a very general way. Okay, and so we talked about the synthase inhibitors, still new research. This is what we're working on, and it's so new that this is all we have. And 26 is pretty questionable whether it's really working or not. Yep, so this is it, basically.
That's all we've got so far. But we do have something interesting. We have a crystal structure, which means, oh, I can't. It's not letting me share my screen. We didn't do the directions. There we go. Dilemma solved. So this is compound 25 bound to the synthase. So right in this dark gray color, that's the S-adenosylmethionine. That's SAM. It's right here. And then the white color, I'll try and rotate it better. This is the synthase inhibitor. And so what's interesting, there's that long acyl tail towards the back. And then something that's worth noting, if I can rotate it correctly, is this. That's a cysteine. And see how close in proximity it is to the end of this acyl tail, which means that potentially if you edited it, you can make it this particular inhibitor into a covalent inhibitor by binding to that cysteine. And so I can also put the surface on just for fun and try and find where I was. There it is. So this is where they kind of join in the back. So there's just the top part of your inhibitor. And then, wow, I lost it. There it is. That is S-enantiomethionine. And you can look towards the back where the dark gray meets the white. That is how the reaction is actually able to happen, because those two pockets kind of join, and then they can do the chemistry and release the autoinducer. So that's how it is able to block that from happening.
Right, and
so more on that, now that we've seen the synthase itself, is how it actually works. So like I mentioned, that S-adenosylmethionine, that was that dark gray compound that was bound in there. Usually the other substrate that goes in is this acyl ACP, and it does this, wow, I just forgot, chart reaction, oh my gosh. Yes, I just like lost the word in my head. But so this is the reaction that happens inside the enzyme, and then this is where that lactinization step happens. It's just an SN2 reaction in order to make the autoinducer, which is kind of ejected, and then this methylthioadenosine is the other byproduct. So this is essentially just what happens, and our inhibitor is trying to block the formation of this autoinducer from being created.
so this whole synthesis is summarized with the BMI1 synthesis, where we have this AHL created. And so what we see here is like we add some extra steps to it, these auxiliary steps with different enzymes, and we have resorphin. So resorphin is able to glow. So the reason why we use this assay is because it's easy to tell if it worked or not based on the glow. So if BMI1 is not inhibited, if it just acts normally, this AHL will be created, the autoinducer will be created, signal can be produced, bad behavior can happen. That's going to mean we're going to have glowing. So glowing is not ideal. We don't want that. If we are able to inhibit this synthase and inhibit BMI1 from the beginning, none of this can happen and we'll see no glow. So no glow, we're good. We have inhibited the synthase and our drug has worked. So this is shown in this very simplified graph. So the more fluorescence we have means the less drug we've used. And the more drug we use, the less fluorescence we have over a period of time. So taking this graph and now using the slope against the concentration of the drug, the more drug we have over here, the less glow we have basically. And again, that's just reiterating ideal conditions that we've done it right, we have inhibited the synthase, and our drug is working. So this is what we're shooting for, and good outcomes. So just bringing it all back together, what we're doing is trying to inhibit the Lux-I synthase, which is the autoinducer synthase of the Vibrio fischeri bacteria. You can see here, so this is JKR, the inhibitor that Julian Kellner-Rogers made to help with this process.
And then what's interesting kind of to note about JKR, if you think back to the synthesis that the actual synthase does, is this is the lactinization transition state, and we are trying to mimic that. So you can see kind of the structural similarities between when SAM is binding to that acyl ACP. That's what they are trying to mimic here with this particular structure in order to successfully inhibit the production of any more autoinducer.
In the end.
It was nice, thank you.
Any questions or anything?
I apologize for not being able to spell.
I just couldn't help myself. The first typo was from the paper. That was a little surprising, actually, that she would do that. But anyway.
Full of surprises.
No, it was nice. I mean, I think the only thing that I would say is that the structure that you showed, I think the best way to think about what that is it's MTA is bound, methylthioadenosine is bound to the protein, which is a product.
Oh,
Then, yeah, so it's not SAN. It's actually it's not the substrate. It's actually the product. And then the other molecule is synthetic, but it looks awfully much like an autoinducer. So what you're kind of looking at in that structure is the completed catalytic cycle, but the products of the reaction are still attached to the enzyme. And that might be just because the autoinducer structure has been altered a little bit. But I don't know if people remember that, but I look at that structure all the time. So that's what it is. And then JKR, I don't know, do you still have Chimera on there?
yeah, I have it pulled up.
Is Chimera shareable?
It can be. Let me do that real quick. Do you want me to pull up that 3P again?
Yeah. Oh, nice. So, yeah, and sort of flick it around a little bit so you can see both those compounds. Yeah, so what our compound does is it actually ours, JKR, has the adenosyl group, the gray group, and the acyl tail group, the white group, and a linker between them. So it binds it binds both pockets through the junction. Yeah. But do people see that the white compound is autoinducer-like? And then the gray compound is the exact byproduct of the reaction, which is methylthioadenosine. So, yeah, so we're trying to make, or at least what we have been doing, is making adenosines with a linker to that long acyl tail to hit both of them. And we have one compound. We have one compound, but one is better than none.
It's true.
All right. Thank you. Well, so next week I'll look at the papers, but Julia and Lindsay will present next, and then the subsequent week, Naya and either Beth or Emily will present, and then the subsequent week, whoever among Beth and Emily didn't go with Naya will go with either Lindsay or Julia. Do you know what I mean? So you'll have a different partner every time. And I think the next paper that would be good would be an enzymology paper of the enzyme. There's a couple crystal structures of the enzyme, and then also, this is actually good for you, Julia, there's like HPLC evidence of how the enzyme catalyzes the reaction. So they just stop it like at points and watch like different intermediates form and so on in the HPLC. So it's a pretty good paper. So, all right.
Scott, are you me and still meeting on Wednesday?
Excuse me?
Are you me and Beth still meeting on Wednesday at 1, like last week?
Yes, we can do that. Yeah.
Okay.
And do you want to send me something before then to look at, writing wise? Or should we talk?
If we could talk, that'd be great because I don't really know where to like start. But
Okay. Yeah. Yeah.
if not, I have time tomorrow, so I can try to get some ideas together.
Okay. Yeah, we can certainly talk. And the other thing that I can do is I can start assembling I don't know. Yeah, I guess maybe what we could do is sort of make a plan and yeah, because you'll need like kind of like a plan and like just some structure and some dates that would be good to have like different sections done. So we can go over that kind of stuff. And and then also maybe some other sort of key papers that you should have in your mind for the background. Although kind of the point I think for just the presentations in general would be it's the same papers like that we'll sort of go through would be like the core background papers that would be helpful to know. So, but yes, we can meet on Wednesday. We'll meet on Wednesday. And then docking people will meet on Thursday morning. Sweet. Nice. Nobody got a haircut? You guys are not allowed to get haircuts now.
I got a haircut not like a while ago. I probably
I noticed when you got your haircut.
yeah, I got it
I
done
think so. Yeah.
mid-August. No, I did. And I like accidentally got bangs and I didn't mean to. It just like happened that way. But
How do you accidentally? It's right in front of your face.
the way she cut it, it's just like much shorter than like the rest of my hair, so like naturally they have become bangs. But I have not been wearing bangs. like your standard
You
bangs.
haven't pushed up. You pushed away.
Yeah, like I would part it or, maybe some tendrils, but no full bangs.
That's funny.
I like on purpose cut bangs, and now they're at like a very weird
Oh, I remember that one.
length, and I don't really wear them, but just look not okay.
I'd be curious about the etymology of the word bangs. Like, what the hell is that? Like, you know what I mean?
Yeah.
It's just like bang right there.
You think that's where it comes from? How do you wear a haircut? I don't know. Bang. You think that's what it was?
Maybe.
Yeah, maybe.
Yeah.
I don't think about that stuff. Gotta be honest. I don't know what to tell you.
I don't know. That could be something else we could work on as a research group. That would actually be hilarious. Imagine like people are like, okay, so like, we want you to give a research talk about like your research over the summer, and you just talk about like the random stuff like that we talk about, like, so we learned like where the word bang, like why do people call bangs bangs? Let's get into it.
On October 6th.
Can we actually?
Yeah, if you want to. I mean,
No.
what's that?
Wait,
Vice actually said yeah.
Yeah, I mean, sure. I doubt that any of you are going to have the courage to do that.
I would.
It makes me want to do it more.
Yeah.
You get a little snippet at the end.
Yeah. I think you should go for it.
Bonus content.
I'm thinking, like, what if we trick them out? Like, we start by, like, very much, like, talking about bangs, and, like, everyone thinks that's what we're going to be discussing, but really we're actually going to talk about, like, what we actually researched. And
I think I think you have to do it at the end. I think you have to do it at the end, because if you do it in the beginning, you could do it in the middle, but I think you can't do it in the beginning.
To have like an
You could just do
intermission.
like an intermission where you're like, Okay, so I'm just gonna take a little break here. We're gonna talk about hair care and hairstyling and such. You know what I mean?
Throw a lot of puns in there.
Oh yeah, for
Just want
sure.
to cut to the chase.
Presentation on quarantine hairstyles
Yeah.
and methods of cutting your hair.
Experimental methods. Have you ever thought about? Yeah, I'll leave that up to you. I mean, you should have creative control. And that's another thing is I think probably it's, have you thought about your presentation much at all? Your actual,
I don't even think we've talked about it.
No, probably not.
because you had, it's like probably about 3 weeks. What day of the week is it? A Tuesday, probably?
Tuesday.
Yeah.
It's probably 2 or, yeah, I guess it's 2 weeks from tomorrow. Is that right? Yeah, it must be. Is that right? Yeah.
Yeah, I think it's the
Tuesday.
6th.
Yeah.
So I would start relatively soon.
Yeah.
What would be great would be to run through it like the weekend before with me and then to time it. And then also if there are things that I think you should change, you have an opportunity to change them. We could run through it even before that too, like maybe one before that and then one the weekend before and then do it. You know what I mean?
Hmm.
How long do we have since we're not doing individual? Is it like 40 minutes or 50?
Well, the maximum amount of time that you have is 50 minutes. So
Okay.
I think you should shoot for well under that.
Great.
Yeah. So it doesn't really matter how long it is as long as you're done in like 45 minutes, right? So that if people have questions, you can answer their questions and then it's over. So yeah. But if it's like 25 minutes, that's fine.
Right.
What are you gonna do? Nothing. It's over. You know what I mean?
Use the rest of the time to talk about bangs.
There, that's
Now we're
how you should do
talking.
it. I'm glad we finished with time to spare because yeah,
Pull up a second presentation on hair.
yeah. So good. Cool. All right. Well, I will see Beth and Emily, I guess, on Wednesday, and then I'll see the rest of you folks on Thursday.
Okay.
All right. Well, thanks. That was really nice. And I'll get the paper to Julia and Lindsay. I'll email you that out in like a couple of minutes. Okay. All right.
All right.
All right.
Bye.
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 Fuck, I never leave on time. God.